Hi, everyone. My name is Caitlin Dreisbach, and I'm a current postdoctoral research scientist at the Data Science Institute here at Columbia University. Today, I'm presenting on my NIH-funded pilot study through the Precision and Symptom Self-Management Center at the Columbia University School of Nursing. This work is titled Clustering and Validation of COVID-19 Symptom Phenotypes for Latina Women. In response to the surge of COVID-19 in New York City, a collaborative interdisciplinary team at Columbia built COVID Watcher, an app to help inform the response to COVID-19 and to better understand the personal and social health implications of the pandemic. COVID Watcher is driven by the approach of citizen science, or the public actively participating in scientific research. Even before the novel coronavirus, women have been reported to share the same medical diagnosis differently than men. The most famous example of this are the somatic symptoms during a heart attack. Though both men and women experience severe chest pain, women tend to present to emergency rooms with nausea, increasing fatigue, and shortness of breath, compared to men who predominantly focus on the chest pain. The core concern is that much of the research underlying the known biology has historically been conducted in men and has contributed to a gender bias in research. My pilot study is employing a mixed methods approach, utilizing the data from 551 users of COVID Watcher who report their gender as female. On this poster, you can see my three specific aims. We do hypothesize that there will be symptom phenotypes based on health status, environmental factors, and access to healthcare that impact a participant's health trajectory throughout the pandemic, and that these phenotypes are different for users who identify as female compared to those who identify as male. Further, we hypothesize that there will be different symptom phenotypes between Latina and non-Latina women. The figure here in the methods section shows the generalized workflow. First, I cleaned and wrangled the COVID Watcher app source data, then performed clustering procedures on the reported symptoms, and then now we are purposefully sampling participants from representative clusters for targeted qualitative interviews and symptom validation. The results I'm showing here on the bottom show a snapshot of the clustering, which represents both aim 1 and aim 2. First, you can see on the bottom left results of clustering from all of the users. Then, as we make our way towards the right, users who identify as male and users who identify as female, and then the bottom two as users who identify as female and non-Latina, and users who identify as female and Latina. And you can visually see a difference, particularly for the users who identify as female and Latina. We further want to explore some of the reasons why this clustering looks different in this group, whether that is because of symptoms that are actually being reported or even just unevenness in the sample. In the synthesis of this work, and after reviewing these initial results, I suspect that the clusters will represent different diagnosis variations, such as allergies, actual COVID-19 or other common colds and viruses, rather than actual COVID-19 condition variation. This is likely a result of the COVID Watcher data itself, where the vast majority of entries are actually none for the symptom attestation question. Our focus here on what is driving this differential clustering in Latina and non-Latina women. Again, while it may be due to a balance of data in the COVID Watcher app, the follow-up interviews are necessary to get more information and a more detailed view of the lived experience. These interviews are currently on track to be completed by the end of April. Thank you for listening, and I'm looking forward to hearing your thoughts on this work.